of lab. Okay, so we're going to go ahead and we're going to start then our new unit, and we're going to be talking about the prokaryotes today. And our lecture objectives are what you see on the screen there. So we're going to talk about characteristics of those prokaryotic cells, and then we're going to finish off with talking about the two domains, archaea and bacteria. So I just want to go back and remind you guys that this word here should be familiar to you, right? Prokaryotes. And so if we remember, the pro here means before, the karyo refers to the kernel, which is the nucleus. So these are cells that have evolved before the nucleus. So prokaryotic cells lack a nucleus, and they also lack all true organelles. Okay, so let's go ahead and take a peek at what a prokaryotic cell would look like. Okay, Okay, there we go. Okay, so let's go ahead and start with the characteristics that are listed in your notes then, and I'm just going to follow A, B, C, and D. And so if we start with A right here, it's going to be a cell wall, and that's going to be then outside of what's called the plasma membrane. So your cell wall, the difference between the two is that the cell wall is very thick and rigid in its structure. It helps to give protection, it helps to give support and structure to the cell. And then just inside the cell wall is this thin layer called the plasma membrane. It is made up of phospholipids there that help control what will go in and out of the cell. You have plasma membranes around your cells, but you do not have a cell wall. But all bacterial cells do have, in fact, have a cell wall. Okay, the next term, cytoplasm, guys, that's not on the diagram here, but let me show you. So cyto, think cell. Plasm is like plasma, which would be liquid. So it's everything inside the plasma membrane, then this liquidy, it's a thick viscous, kind of like a thick Wesson oil here, in which all of the internal components of the cell, they're floating around inside the cytoplasm. So prokaryotic cells have cytoplasm. In addition to that, your prokaryotic cells also have these little dots right here. On my screen right here, they kind of look like little kind of brown mustardy dots, and those are ribosomes. And ribosomes are, by definition, not a true organelle because they don't have a membrane around them. But what they do, guys, is they are there for the synthesis of protein. So protein production happens in the ribosomes. And then in order to be able to know what proteins to make and how to make them, when to make them, and all that good stuff, then the cell is going to have instructions here in this circular chromosome. It doesn't look circular here because it's kind of overlapping on itself. But that would be the circular chromosome, different than ours because we have linear chromosomes, but similar in the fact that it's still made up of DNA, but they have a single circular chromosome. Okay, so those are the standard everyday structures then for a prokaryotic cell. So if you move down in your notes then to number 2, it says optional structures for prokaryotes. So in no specific order here, I'm just going to follow what the paper says, and then most of those are going to be the highlighted labels that you see on your screen. So let's go ahead and start then with this one right here, the capsule. So the capsule then is going to be the layer, if it exists, that's just outside the cell wall. And the capsule is very sticky and slimy. It's almost jelly-like in its substance. It allows then for the cell to adhere to members of other cells within its population. It can allow for attachment inside a host, like onto their tissues, for example. And the capsule also allows for protection. And so if we go here for just a second and we look, this is also a bacterial cell. It's just a different shaped bacterial cell. That would be the cell wall right here. And then outside of that, there's that bacterial capsule. And you can see that that is allowing then for attachment to the host, which right here, as the diagram shows, is a tonsil cell. So if you've ever had strep throat, then this is a strep infection, and you get sick because that bacterium attaches itself using the capsule then to your tissues there. Okay. The next on the list are the pili or pili. And your book gives you a specific type of those structures, this one right here called the sex pilus. And so the sex pilus, as you can imagine by the name, this is going to be used for quote unquote bacterial sex, a form of sexual reproduction in a way. I'll come back to that in a little bit and explain what I mean by that. But out of all of these little finger-like projections, the one that is here that's a little bit longer than the rest, that's going to be the sex pilus. And what the sex pilus is going to allow for then is the movement of genetic material from one bacterial cell to another. And that's going to happen in a process that's called conjugation. Again, I'll come back to that a little bit later. I think I have a picture of that here. And so this would be, for example, the donor bacterial cell. This is the recipient bacterial cell, and sandwiched in between them then is the sex pilus. Think of it as kind of a long tube that physically connects this bacterial cell to this bacterial cell, from which genetic information can be moved from one cell to the next, from the donor to the recipient. Okay, let's see what's next. Fimbriae. Okay, so we've got these fimbriae right here, and you can hopefully see that they are quite numerous. They are shorter than the sex pilus, or these things here called the flagella. So they are finger-like projections that, in this case, are coming out of the capsule. And like the capsule, they are also there for attachment. And so if you've ever heard of the infection gonorrhea, it's a sexually transmitted infection. That's how this guy works. It's a bacterial infection that has the fimbriae on the outside of its capsule, and it does a really good job of attaching itself then to you, the host. And again, that's going to be transmitted sexually. Okay, we have this next one here. Flagella is plural. Flagellum would be singular, but many of the bacteria will have the flagella, and this is basically a structure then that's going to be used for locomotion. And for locomotion, the fancy term, guys, is taxis, spelled T-A-X-I-S. So think of taxi. If you want to go from point A to point B, you might hail a taxi to get you there, to move you there. Here, the flagella is allowing for movement within the bacterial cell, and that again is called taxis. And I believe I have a picture. There we go. 27.7 is showing you then what the mechanics, the overall structure of the flagellum would look like. And so this one, it works a little bit differently than the flagellated cell that you know as a sperm cell. This would be a really good example of an analogous structure because the bacterial cell did not give the eukaryotic cell the flagellum. Rather, they evolved independently. And we know that because they look very different from one another. So this type of flagellum here on the prokaryote is kind of like a toy, like a jack-in-the-box toy. So it's kind of like a crank. So it's, you take and you spin it rather than a whip-like type of flagellum, which is what the eukaryotic cell has. So this one, as you can see here, it's just showing you that there's a rotation happening in the top of the flagellum, allowing this then to kind of have a corkscrew type of movement, allowing then for taxis. Okay. Some of the bacteria will have an endospore, and then some of them will have a plasmid, neither of which are here. So I have some slides here. So there's the fimbriae. Again, that's for attachment. And then this is going to be the endospore. So if you notice, this is going to be then basically the bacterial cell with its genetic material that has shrunk down and kind of dehydrated itself, if you will, to a very small portion called the endospore that has a coat around it. And what the endospore is, it's a dormant cycle or a dormant time period within the life cycle of the bacterium. So if, for example, the bacterium finds itself then in an inhospitable environment, then it will produce an endospore, and that allows it to kind of overwinter, become dormant. It's very tough. It's non-reproductive, but it does in fact protect that bacterial cell from environmental stressors. And some of them can withstand high heats up of about to 121 degrees Celsius. Anything above that usually would destroy the endospore, but it works really well in protecting that bacterium. Okay, And then the last optional structure, that's 2F here, this is going to be a plasmid. And so as you can see here in figure 27.9, all of this kind of spaghetti mess right here, that's the circular chromosome. But if you look over here, this little kind of rope that looks like it's also in a circle but overlapping itself, that's actually the plasmid. It's also circular, and it is very small, and it carries a very small amount, as you can obviously see by the size, of genetic material. And these things can be pretty scary because the genes or the genetics that they do carry often code for genetic advantages for the survival of the bacterium. So worst case scenario, for example, is that the plasmid could carry genes to code for antibiotic resistance, which we've talked a little bit about in class. And so if you are unfortunate enough then to be hit with a bacterial infection that carries a plasmid that is antibiotic resistant, then it could be that many antibiotics don't work, and it could turn into an MDR type of infection. So just to be clear, the chromosome here, that's, remember, a single circular chromosome, that's going to code for just your regular daily functionings, while this over here is going to be a specific type that has nothing to do with daily functions. Okay, so that takes us then through the structures for the prokaryotes. So the next thing that I want to do, guys, is I want you to go to B, prokaryote classification number 1, where it says 3 common shapes. So if we could go take a peek at that, let's go there. Okay, so this is going to be one of the things that your lab is going to ask you to do. If we were still meeting face-to-face, you would have a single slide with 3 different samples on that single slide, and those 3 samples would be representing what you see here on the screen in figure 27.2. So there are 3 common shapes that we're going to find in the bacterial world, and we'll just start with A. So I left some space for you in your notes there if you want to go ahead and maybe give yourself a quick drawing of it and then name it. So A, I'm going to amend some of these shapes. Don't want to refer to it as spherical, but these are going to be cocci, C-O-C-C-I is how you spell that, and these are going to be circular shaped bacterial cells. Okay, cocci, C-O-C-C-I. If we move over to the center, to this right here, these are often referred to as rod-shaped, but the true scientific name would be the bacilli, B-A-C-I-L-L-I, B-A-C-I-L-L-I, and those are your rod-shaped. And then this one over here, this one is spirilli, S-P-I-R-I-L-L-I, and they have a spiral shape. As you can see, they look very corkscrew, and almost kind of like a little worm going on right here. The ones that we have on our slides in class aren't as long or are as spirally, but look kind of more like a very sloppy S. And the spirilli, this is one that you may have heard of before. This is going to be the cause of Lyme disease. So if you're out doing a hike here soon and you're going to go look at the wildflowers, you might want to wear light clothing like white long pants, shoes, long sleeves, so that you can see if a tick does make its way onto you. A tick is an ectoparasite that could possibly vector Lyme disease, and if they were to get a blood meal from you, they have then the possibility of transferring and vectoring this kind of spirilli type of bacteria. If we go back to the middle for just a second, your rod-shaped, these are going to be things like E. coli. So I'm sure that is a bacterium that you guys are very familiar with. And then if you go to A for the cocci, that would be something like a strep right there. Okay. All right. Now, one of the things that we did before we left was we swabbed our hands, and we were doing that in anticipation of doing gram stain. And so that's going to be part B, number 2 here. And I'm going to take you to a screen to show you then what gram stains do and why we do it. So what you guys would have done is you would have removed some of the bacteria from the pure cultures from the plates that you guys made last week. And you would have gone through a series of steps of staining, which I'm not going to bother going through now because we're not going to do it. But basically, this is it in a nutshell. So when you do a gram stain, you can have one of 2 options or 2 two findings here. So gram-positive bacteria is one result, or gram-negative bacteria would be another result. And this is a test that has been around for a very long time. In fact, 1884 by Hans Christian Gram, hence gram stain. And it's quick, it's cheap, and it's easy to perform, so that somebody in the laboratory, or your doctor, for example, would know which type of bacterium they're looking at, gram-positive or gram-negative. And what this is saying is this is giving us a lot of information without even knowing the specific species, but a lot of information about the cell wall composition. So it could look something like this or like that. And if it looks this way as it does in a gram-positive versus this way in a gram-negative, then that's going to allow then people in the medical industry to determine how to treat these, because different antibiotics will work differently on gram-negative bacteria than they will on gram-positive bacteria. So if we go and we look, let's take a peek at this side over here. We'll just start with the gram-positive. And so if you look at this layer right here, the cell wall layer of the peptidoglycan layer, okay, it's very thick, and this is a very rigid structure. Underneath it, that's the plasma membrane that I was talking about. And so this is a very lipidy kind of oily substance covered by a very thick, rigid structure. If I move over here to B, you still have that layer of the peptidoglycan, but if you notice, it's much, much thinner, and it's now sandwiched between two membranes, the plasma membrane and then the outer membrane, again, very lipidy. And so when you go through and you stain, and then at the end you wash the stains off, you're either going to end up with a gram-positive bacteria that will have a very deep purple color to it, or you will have something that has a lighter pink color to it. And in the gram stain for a gram-positive bacteria, when you put on purple iodine, it is then going to basically be soaked in, if you will, or absorbed by this very thick layer of peptidoglycan. And when you go to rinse as your final step, the purple color remains. If over here you do the same layers, the same stains, I don't have a thick layer of the peptidoglycan to absorb the stain. That stain is going to go over this outer lipidy slimy layer, which will then wash off. And so that makes then this guy here look very dark purple, like you're seeing here, that are labeled the gram-positive bacteria. And if you can tell the shape of those would be the cocci. And if the results are giving you a gram-negative bacteria, then they're going to look like these guys here, which are lighter pink color, because you put a saffron layer. If you didn't put a saffron layer, then there would be no color here. So the saffron actually sticks to this membrane layer, giving it the pink color. And that, if you can tell, those are going to be the bacilli, or the rod-shaped. Okay, so again, in a nutshell, the importance of this test and what you need to know is that it's a quick way to know if you have a gram-positive or a gram-negative. It then automatically allows you to know what cell wall composition is there or not there, which will then allow you to go through and hopefully be able to prescribe the proper antibiotics. Okay, we're going to move right along, and we're going to talk about then more characteristics for bacteria, and we're going to look at metabolic diversity. Okay, so here, let's just go to the notes for just a second. In metabolic diversity, we're looking at trophic or feeding categories. Now, the term trophic that's here in your notes, right, guys, that should be already very familiar to you, because we've looked at autotrophs and heterotrophs. We've looked at trophic levels within the energy pyramids and food webs. So now we're going to look at different ways that the bacteria can feed themselves. And remember, bacteria have been around for billions of years, and they've been quite successful. They're ubiquitous, which means that they are everywhere. So they kind of know what they're doing. And so one of the ways that a bacterium could feed itself is via what is called photoautotroph. That's no different than the autotroph that we've looked at, but they're just putting photo in front of it to indicate that they're using light. Okay, so they literally are light self-feeders. And the example that I have up here on the screen, this is a micrograph, one that you'll be looking at for this lab, is called oscillatoria. And you can see that it's green. And it has these lines here, and these lines then are going to be cell walls. And the green stuff in it, they don't have true chloroplast because remember it's a prokaryotic cell. So they just have the some membrane with pigment, but not true chloroplast. And there's one cell attached to another cell, attached to another cell, attached to another cell. And so this is then a colonial type of bacterium, if you will. Okay, that you don't see individual cells stuck together. And these guys and other types of photoautotrophic bacterium that have been around for 2.5 or more billion years, they are responsible for what is called the oxygen revolution. And we should be thankful to these guys because they are the ones then that first evolved the ability to take in CO2, convert sugar, and then release oxygen as a waste product, and molecular oxygen, the way that, or the form rather, that you and I breathe this in. And so when the Earth was first forming 4 billion years ago, 4.5 billion years ago, there was little to no oxygen, which was a good thing because oxygen actually likes to rip things apart. But then along came these guys a couple billion years later, and they're just pumping out massive amounts of oxygen, then paving the way for life as we know it today. And I'll show you this again when we get to our lab intro. Okay, we also have what are called chemoautotrophs. So again, that's one reason why you have to put the prefix photo or chemo. So for B, chemoautotrophs. So what a chemoautotroph is then is going to be an organism that produces its own carbon-based molecules using chemicals, carbon, nitrogen, for example, not using light, but they're still making their own. Sugars, guys. Okay, so here is an example of a really spectacular type of bacterium that kind of fits both definitions, photoautotrophic and chemoautotrophic. And the genre here is Anabaena, and you guys will be looking at this and drawing a picture of this in the lab that corresponds with this lecture. And what's really cool about this is that it can feed itself with or without light. So if we go and we look at the cells that are listed here that are photosynthetic cells, okay? These photosynthetic cells are just referred to as vegetative cells, which means that they're going to photosynthesize, just like the name implies, as long as there's light. But imagine if this organism in the water finds itself in an environment that has no light. If it's photoautotrophic and it doesn't have enough light, then obviously it would die. But this one doesn't do that. It has these other cells, so there's one right there that's labeled a heterocyst, and then there's another one right over here also labeled a heterocyst, and these are going to be the chemoautotrophic cells. So let's just back up for a second. You have a colony of bacteria, filamentous bacteria, that are all stuck together. Most of them are vegetative cells that are going to feed through photosynthesis, while others in the colony are going to be these round cells. You can kind of make out that they look different than the vegetative cells, called heterocysts. And they're going to undergo nitrogen fixation, and that is a type of chemoautotrophic metabolism. Now, the nitrogen fixation should not be anything new. We talked about that with the nitrogen cycle, but just to review really quickly, remember that much of our atmosphere is N2 in a gaseous form with 3 covalent bonds between it, which we cannot break apart. But organisms like these heterocysts and other bacteria are nitrogen-fixing bacteria, and they easily can break apart then the triple covalent bonds within the between the two nitrogen atoms there. That's what these guys are going to do. So if we go back to Anabaena, and it finds itself then in an area that's lacking light, then they'll switch their metabolism to being chemoautotrophic, and those heterocysts will start to undergo nitrogen fixation. So they will use nitrogen then instead of light to make their own food source. Pretty cool. Now, another thing I want to say about the heterocysts really quickly before I move on then is that these guys are kind of flotation devices in addition to being nitrogen fixers. And the other thing that's interesting about them is that they have a weak point of attachment. So here and here, here and here. And so these are really good at asexual reproduction. And so if they just kind of detach themselves there and maybe there, for example, then this chain will, or this filament rather, will go through asexual reproduction and make more of itself, as will that chain. So in addition to fixing nitrogen, they help to keep everything afloat, and then they're weak attachment points, which is good for asexual reproduction. Okay, and then the last one that we have are chemoheterotrophs. And I don't know if I have a slide here. Let me just check really quick, guys. This is just a picture of the chemoautotrophs. So these nodules here are going to be filled with these nitrogen-fixing bacteria. So that's just another example similar to what we saw in the Anabaena, but on plants. Okay, I don't have a slide for chemoheterotrophs. So let's just define it and then we will move on. So chemo is chemical, hetero is other, troph is feeding, right? So these are going to be bacteria that you have, for example, in your gut. And they're going to be living off nutrients that you provide to them. They cannot make their own food source whatsoever. Many of the infectious bacteria, those that are pathogenic or parasitic, they are going to be examples of chemoheterotrophs as well. Okay, so just not able to make their own food whatsoever. Okay. Moving on then to oxygen and the use of oxygen, or the role of oxygen in prokaryotic metabolism. So we have obligate aerobes, obligate anaerobes, and then we have facultative anaerobes. So if you think of the terms obligate, it sounds like obligatory, right? And that's something that you have to do. So if we go to the next screen here, there you have pictures of test tubes with red circles, which are supposed to be the bacteria floating in a specific spot in the test tube in the water. So obligate aerobe, obligate anaerobe, and then facultative. Okay, so let's just first start with the term aerobic and anaerobic. Aerobic, that term then is something that requires oxygen for survival. You and I, for example, we are aerobic organisms. We have to have oxygen to survive. That's not true of all organisms. If we go and we look at the term anaerobic or anaerobes, these guys right here, these are going to be organisms that do not require oxygen, and in the extreme cases are actually poisoned by oxygen. Okay, so aerobic requires oxygen, anaerobic does not require oxygen. So if we go and we look at this one here, obligate aerobes, if you look at the bacteria, you're going to notice that they are growing and concentrated only in the top portion of the test tube where they can get oxygen. Okay? If you go to the middle where it says obligate anaerobe, those are in the complete opposite spot of the test tube, floating down at the bottom because they absolutely cannot have oxygen. If they find themselves in the presence of oxygen, they will die. If you go over here to facultative anaerobe, you see that they are concentrated here at the top like they are for the obligate, in the middle, and some of them down at the bottom. So maybe you can see from that picture right there that if it's a facultative anaerobic type of bacterium, that it actually prefers to not have oxygen, but does okay if it does have oxygen. So kind of like the feeding. I can feed if I have light. I can switch and feed in the dark if needed. Here, they could feed if it's facultative in the presence or not in the presence of oxygen, and they're perfectly okay. So guys, just kind of appreciate that this is, one of the reasons why the bacteria, why they are so successful, because they have all of these mechanisms that they can put into play if the environment changes, or if the environment isn't just right. Okay, so I would imagine that that bacteria can grow really fast. So bacterial growth is fast and efficient. Some of the fastest bacteria, like E. coli, if they're in a perfect environment, they can actually reproduce themselves once every 20 minutes. And so within a time period of 12 or 24 hours, you can easily get a population of bacteria that are well into the billions. And one of the ways that they can do that is using asexual reproduction, which specifically for this group is called binary fission. And if you're not an E. coli, but you're more of a typical bacterium, it takes 1 to 3 hours to go through the process from start to finish. Okay, so if we are looking at binary fission, bi is 2, right? Fission means to split. So that's essentially all that's happening. So it's a very simple process. So if you look at this bacterial cell right here, one of the things, like all cells, that has to be done is that it has to duplicate all of its DNA. So after DNA replication occurs inside the cytoplasm, you see that we have a cell that's a little bit longer, and you can see that there's a longer kind of mess of DNA right here, a messy chromosome. If we continue the process here, then that chromosome, or those chromosomes I should say, because we've gone through replication, are going to start to migrate to either sides of the cell, like they have here and here. You can see that the plasma membrane and the cell wall there and there are going to start to invaginate on themselves, or kind of pinch towards one another. And this process called cytokinesis, the splitting of the cytoplasm, occurs, leaving us then with 2 identical and completely intact bacterial cells. And again, 1 to 3 hours average is what it takes for that to happen. As you can imagine, then those 2 cells that are there, they can each also then go through binary fission, and that's going to end up giving you 4 cells, and then those 4 cells to 8 cells, so on and so forth. Because they prefer asexual reproduction, you don't have to find a mate, and it's quick and it's easy. And because it doesn't take very long for them to reproduce themselves, they have short generation times. You're going to see that evolution is going to evolve very quickly in a population of bacteria, much more than it would ever in an animal species, for example, because they're just sitting there pumping out new copies of itself. And every time the DNA is duplicated, there's a chance then that a mutation will occur. And if that mutation is not corrected, then that mutation carries on, allowing then for that species obviously to evolve. Okay. But sometimes if an environment is inhospitable, then the bacterium will need to actually go through sexual reproduction. And the first type of sexual reproduction that we're going to talk about is what is called conjugation, and that's where that sex pilus comes in, the sex pilus that I showed you earlier in the start of the lecture. Okay, so you've got 2 different figures going on here. So the figure at the left is showing you and giving you words then of what's going on. Don't get bogged down with the HFR and those labels, but I just wanted to show this one because it's giving you different colors. So if we look at this circular chromosome here and that cell, which is the donor cell, and the chromosome here and the recipient cell, you're going to notice that some of the DNA in the donor cell is going to be replicated and moved through that conjugation tube, which is the sex pilus, and incorporate itself into the receiving DNA, or the receiving cell. If you notice, after the DNA is copied and moved, or partially copied and moved over, the chromosome here looks just as it does at the beginning here, but this chromosome here, once it incorporates, has new genetic information in it. And so that's going to allow for genetic variation, which is going to be quote-unquote sexual reproduction, not asexual reproduction. And so what you see going on here then is the donor cell, the recipient cell, and these right here are going to be the fimbriae, okay? This is your sex pilus, and so a small portion of the DNA that's been copied from this circular chromosome will move through that sex pilus. It will incorporate itself into the chromosome that's in the receiving or the recipient bacterial cell. When this bacterial cell receives its information and looks genetically like it does here, which is different than it was in the beginning, then it will probably decide to go through asexual reproduction, or that binary fission, and just continue to make more copies of itself. And voila, there you go, right? You have evolution that has occurred, and it didn't take very long for that to happen. One thing I want to point out before we move on to the other types of genetic exchange that are possible is the fact that this bacterial sex, if you will, is very different than sexual reproduction in animals and in plants, for example. because you don't end up making any offspring, right? All you're doing is transferring genetic information, and then that bacterial cell has to go through asexual reproduction to make copies. But there's no quote unquote offspring that are coming. So this I once read, and I thought that this analogy works well. Imagine taking your iPhone and plugging it into your computer using a USB cord, trying to get music from maybe iTunes from your computer onto your cell phone. The information is going to travel through the USB cord, i.e. the sex pilus for bacteria. And when the process is done and you detach it, you don't have a bunch of new Apple products, which would be awesome, but you don't. But the information that's on your phone then, after the transfer of information, is now different than it once was. Okay, so hopefully that makes sense. Okay, some bacteria will go through what is called transduction. And transduction then is using a virus instead of a sex pilus, but a virus then to transfer DNA from one spot to the next. So if you are looking at the top here from figure 2711, this right here is going to be your bacterial cell. This kind of funny looking injector thing, that's going to be the virus. Okay, that is specifically referred to as a bacteriophage, but don't worry about that. Just know that that's a virus, and the blue in it is its genetic material. And as you can see here, the virus attaches to the outside of the bacterial cell, injects then its DNA. Okay, when that happens, then this information is going to kind of take over the bacterial cell. It's going to cause then disruption of the circular chromosome that we see that's intact here. It's going to break it up, and it's going to cause then this bacterial cell, instead of doing what it needs, to now make more viruses or bacteriophages. But if you notice, out of the four bacteriophages, one of them is different. One of them is carrying some of the genetics from the bacterium rather than the virus. And so this then is going to be released out of the bacterial host cell, because the bacterial host cell explodes when it's filled up with these virus particles or viral particles. So they're all released, and if we follow this one, it's going to go and infect a bacterial cell, and it's going to inject its DNA. And if you notice the A plus here, that's just trying to show you that that's a piece of genetic information from the original bacterial cell. And that DNA then comes in from the original bacterium and inserts itself into the new bacterial cell, replacing the A minus with the A plus. And so when we look, when it's all said and done, then this is a recombinant cell. So all that means, guys, is that this bacterial cell right here, its genetic information here is different than it was at the start. Instead of being A minus B minus, it's now A plus B minus. And so it has kind of the same result as conjugation, but instead of using another or a second bacterium and a sex pilus, the same goal is achieved by using that bacteriophage or viral particle. Okay, and then finally we're going to move to the last one, and this one is really scary to me, that this is even possible, and this is called transformation. So what you have here is you have, again, your recipient or your receiving bacterial cell, and then in the environment you just have random pieces of DNA. Okay, and this could be DNA from dead cells, dead bacterial cells that's cut from different fragments. In the environment, it could be cut DNA from other species. Again, that's what can make it so scary. And if you notice that what happens is that the DNA in the environment is going to be picked up and brought into the cell here. So this bacterium, the recipient cell, picks up some random genetic information from the environment and brings it into the cytoplasm. Now, as long as it's genetically compatible enough, so if you see the genes or the genetics here are listed A, B, C, and D, they're listed here A, B, C, and D as well, so they are compatible. It's just that this here is a different version of the genes because they're uppercase. This here is a different version because it's lowercase, but nevertheless, they're using the same letters, so they are compatible. And if you see, a small part, totally random, is going to be incorporated here into the original circular chromosome. So instead of having capital A, B, C, and D as its genetics, this bacterial cell now has little a, big B, big C, and big D in its chromosome, changing its genetics. And that could be something that doesn't really matter, could be kind of a neutral type of mutation or change. This could be something that allows for the bacterial cell to become more virulent, which means that it's going to be more disease causing. It could have a negative impact on the survival of the bacterial cell. It's a complete crapshoot, but the point here, though, guys, is that the DNA is transformed within the bacterial cell simply by picking up random pieces of DNA from the environment. Okay. Do you imagine if you could do that, right? Just go pick up pieces of DNA and eat it and see how that changes you. It's just kind of a crazy scientific thought, or sci-fi rather, type of thought there. Okay. So we've gone through, it looks like, most of the characteristics that we needed to get through. And so what we're going to then finish up with, let me just check the notes, is we're going to finish up with very brief talk about the domain archaea. We're going to look briefly at the domain bacteria, and then we're going to wrap it up with the fact that not all bacteria are bad. We've got many, many, many beneficial prokaryotes that we'll wrap up with. Okay, so if we just jump back up to the domain archaea, if you remember this, the archaea are in fact prokaryotes, but they are the prokaryotes are the bacteria that have been around for billions of years, and therefore they were evolving when the environment was very extreme and very different than what it was now. So they are called extremophiles. Extremophile then would be something that is found only in extreme environments. So I'm going to just do a quick, okay. So the slide here, if you are looking at a prokaryote and they've described it as being an extreme halophile. So let's look at this. The halo here is salt, and then the phile here comes from philic, and philic, P-H-I-L-I-C, means loving. So literally salt loving, okay? And these are going to be found in extremely salty conditions. Some of them, in fact, have to be in conditions with a salt concentration higher than 9%. Most of them are going to be found in areas that are much higher than that, say 30% salt solution. And you would think that they wouldn't be able to survive because it's way too saline, which would mean then that the water would leave the bacterial cells, causing them to die, but they have specific and special proteins to allow them to survive in such salty conditions. Extreme thermophile, so thermo is heat, phile again is loving, so they are going to be found in very, very extreme, very hot conditions. Not uncommon for these guys to be in areas above 252 degrees Fahrenheit, and you would think, well, that's too hot. We've talked about homeostasis and regulation, and that if you get a little bit hot, right, with the fever, your proteins change and they don't function properly. Well, somehow, some way, they have managed then to mutate themselves so that they're able to tweak their bonds that hold the proteins together and keep them together even in these very extreme and hot conditions. All right, if you go over to some of the swampy areas, if you're ever going to go out to like Louisiana or areas like that, you might then find extreme methanogens, and these are going to be in areas that have very little to no movement in their water, and then therefore little to no oxygen. And so they are in swampy and marsh areas, and you can see that they are strict obligate anaerobe, and remember the obligate is obligatory, anaerobe is without oxygen. So these are going to be organisms that are going to be found kind of in the deepest of the swampy waters where there's no oxygen whatsoever. And you would think, wow, there's no way anything could live there, but they actually thrive in those kinds of conditions. And then to finish up for this here extreme thermoacidophile. So if you take a moment and you think about what that word is, right? Let's break it down. Thermo is heat. The acid would be very acidic and loving, so very hot and acidic type of environments. You will have the thermoacidophiles. Okay, so the good news for you guys is this group here, the archaeans, those organisms in the domain archaea, we don't have anything to look at in the lab, and I just like to introduce you to these. I want you to know that they exist and that you are familiar with the terminology that we use. Okay, now moving on to the domain bacteria. So this is where we're going to spend most of our time when you guys get over to the lab part that I'll be posting here shortly. You'll be able to go and you're going to be looking at organisms that are in the domain bacteria. And there is one table at the beginning of the lab that asks you to list the domain. And so the domain, I'm giving it to you here, is bacteria because I have nothing to show you in the lab from the domain archaea. So bacteria have many different type of ecological interactions. They can be mutualistic, as shown here on the screen. I'll come back to that in a second. They can be commensalistic, and they can be parasitic or pathogenogenic. So if you're looking here at this one, this is what is called a flashlight fish. And the flashlight fish has this big spot here by its eye, and there are billions, or maybe not billions, but lots and lots and lots of bacteria living in this area right here. And they have the ability to bioluminesce. And so in the presence of danger, for example, or if there's a potential predator coming, then the bacteria, they'll all start to flash simultaneously, kind of like a flashlight, and that will confuse, hopefully, the predator, causing the predator to go away. And in turn, because it's a mutualistic relationship, right, the bacteria need to get something out of it, and they get a place to live and food. Another type of mutualistic bacteria that I can think of, you've got E. coli in your gut. There are many different strains of E. coli, and there's one good strain that we need. And right now, as you're sitting there listening to this, right, you've got a strain of E. coli that is busy making vitamin K for you right now inside your gut. In fact, you have more bacterial cells in the gut than you have cells that make up your body. Okay. I don't have a slide for commensalism, so let me just give you an example of that. So you guys have probably been using a lot of hand sanitizers lately. You've probably been washing your hands like crazy, right? And you know that you need to do that because there are bad bacteria on your skin. But there are also bacteria that are on your skin that don't do anything bad or good to you, and those would be commensalistic. So these are going to include species of bacteria on your skin that they basically live off of eating the oils that your skin secretes, and you can have on average 10 million of these bacteria per centimeter squared, so not in a very big area, and you've got a lot of bacteria there. Most of the time they will stay as commensalists, where they benefit and they don't help or harm you. Sometimes though, if you get an infection or if you get an opening in the skin, then they can become parasitic, but usually commensalistic bacteria are just there feeding off of your oils, and that doesn't help you nor does it harm you. Okay, parasitic, this is what I was talking about before. So if I were to ask you, for example, on an exam or a quiz later, give me the shape of this bacteria, you should be able to tell me that that's spirulli. And the organism that's here that vectors this is the tick. And you can see then that tick gave this person Lyme disease, and so this spirulli bacterium found its way into the body. And this is a very indicative and typical type of symptom, or sign rather, that a person might have if they have Lyme disease. It's the very target, what's called a target rash, but not everybody gets that, so sometimes it can be hard to actually diagnose. Okay. Oh, I'm sorry guys, I had this out of order. So there's your commensalistic bacteria. Kind of a cool picture, right? Here's your skin, and then those would be the cocci-shaped bacterial cells that are living on your skin, just basically feeding off of your oils. Okay, back to the parasitic for just a second, and I'm sorry, I can't pause this If I do, I have to do major edits, and so I'm just trying to get through this, so kind of bear with me here. But back to parasitic. If it is a parasite and it causes disease, then it is more specifically referred to as a pathogen. So you might have heard of, I'm sure, many of these. Botulism is a disease, right, that infects the base of your nerve cells, and you can get this from eating food that's not been preserved properly. Many years ago, people used to put honey on the nipples of bottles for babies and to get them to take the bottle. And there was a bad batch of honey that had botulism in it, and it killed babies, so you don't do that anymore. If somebody is, during the holidays gives you, I don't know, canned jelly or canned fruits, and you're not sure they know exactly what they're doing, then maybe be polite and say thank you very much, but maybe don't eat it just in case it wasn't preserved properly, because it can cause some major problems there. Cholera, this is going to be an intestinal infection, and you normally are going to get this in areas, many of the developing worlds have this because their water is not sanitary, right? So it's in contaminated water. The endospore that I talked about before, cholera often makes endospores and can live in the water for years and years and years until it reaches the inside of your body, for example, where it will become metabolically active. Tooth decay, you're supposed to go to the doctor, right, and get your teeth cleaned and the plaque and all that good stuff scraped off. That's going to be a pathogen, a bacterial pathogen. So fun stuff, gonorrhea and chlamydia, we've talked about this a little bit. High rates of those in Kern County, so lucky us. And as you know already, I hope, that is a sexually transmitted infection that can cause infection in the urethra or the fallopian tubes, and they can get in there and they can attach themselves, for example, using the fimbriae. And so you have to have an antibiotic to clean them out from the urethra rather than just having the urine clean them out for you. It's not strong enough to do that. So chicken, undercooked food and things like that can give you other problems in your intestine. That would be from salmonella. And then I showed you earlier the strep throat picture with the cocci type of bacterium with a capsule that was stuck to the cell in the throat. So lots of bad things going on there, but lots of good things as well. So we have CRISPRs. Maybe you've heard of this before. If you haven't, and when you guys get over to Bio 3B, I believe that you read a book now on the CRISPRs. But CRISPRs are short repeats of DNA segments that are used to recognize and cleave DNA. And that's coming from bacteria, and that allows us to then edit genes within organisms. So you can basically use the bacterial DNA to go in and cut out bad genes and put in good genes. So that's the thought. We'll see where that goes. Decomposers. The screen here is not showing that, but if you go to your notes here for number 2, decomposers, so you know already, right, that we have to have decomposers. If we didn't have decomposers, then the whole idea of chemical recycling and geochemical cycles would come to a screeching halt. We have to have decomposers that are breaking down organic materials and helping to replenish those within our soils, in our water, in our air, etc. This here is showing then bioremediation. And so what they've figured out that they can do is, in the event of an oil spill, for example, you can spray the area with these bacteria. And these bacteria, as you can see inside, right, these are oil pockets. So they go in and they metabolize the oil. So what they've got going on in the sprayer then is they're spraying the oil with these bacteria, and that's what they're going to metabolize and then clean it all up. And then some people say, well, what happens though? You've got all this bacteria in the environment, but they should die after the oil is all metabolized, because they no longer would have a food source. So no need to panic there. Nitrogen fixation, we've talked about those. Synthesis of food, hormones, and antibiotics. And then, as I mentioned, to finish it up here, the bacteria, we should be thankful for them because they are continuously putting oxygen into our atmosphere along with the plants. Okay, so that takes care of that. I hope it wasn't too painful.